In this lecture, I will discuss about the application of bioinformatics, right, in computer drug design. We have studied about various techniques in bioinformatics using sequences and the structures, and also we discussed about various types of interactions and how this information can be used to identify the lead molecules or the probable inhibitors, right, for any specific targets. Because in the current scenario, it is very important for the development of drugs, because it takes about 10 years from the beginning to come to the market, and many lead compounds they fail and various different clinical trials, right. In this case, it is very important to have a set of molecules, right. It could be lead compounds, right, for initial start. So, I will discuss different techniques, for example, the docking and the virtual screening and quantity structure activity relationship, right, to identify the probable inhibitors, right, generally used in computer drug design. In the last few classes, we discussed about the proteins, protein folding rates, protein stability and protein interactions, and specifically last class we discussed about the protein interactions, right. What are different types of complexes? Protein complexes, protein nucleic acid complexes, protein carbohydrate complexes and protein ligand complexes, right. And all these complexes they are important for several functions, right. So, and we can identify the binding sites or the interface residues for any complex. So, we discussed about 3 different criteria to identify the binding sites. What are 3 different criteria we discussed? Distance based criteria,
change in solvent
based on solvent accessibility,
energy
as well as interaction energy, right. Using this information, we identified some residues which are preferred to be the binding sites and the binding partners, right. Between 2 proteins there are some residue pairs are preferred and they are involved in different types of interactions, right. We discussed about 3 different types of interactions, right. What are different types of interactions we discussed? Aromatic interactions, right, and cut and pi interactions and electrostatic interactions, right. Then to understand the importance of these interactions, we need to see whether the mutation of these specific residues at the binding interface change or alter the binding affinity, right, with the respect to other residues. So, we discussed about a database, right, called the Proximate, right, which contains information regarding the binding affinity upon mutations, right. This can be a useful resource, right, for researchers to understand the binding affinity, right, of any protein complexes upon mutations. So, when you go to the protein ligand interactions, right. So, what do you mean by ligand? What is the meaning of ligand? Generally we can say any small molecules, right, ligands are small molecules, or you can say any substance or any chemical compound, right. It can be a drug or any function group that bind specifically to any other entity to make a complex. For example, a small molecule that can interact with a protein like a protein ligand complex, or this drug molecule can interact with the DNA like the DNA drug interactions and so on. Here I show one example, this aspirin, right. What is aspirin? Right, it is a painkiller, right. So, in that case if you have a headache, then you can use have this aspirin, right. This can say is a drug. So, if you do look at the protein ligand binding, so you have protein and you have ligand, if they binds, the ligand is usually a signal triggering molecule, because this will go and attach with the pocket in a protein, right, and this will trigger, right, at the binding site. So, where this will target, right, at the protein, that site is called the binding site, right, in a target protein. So, I show this ligand binding to any receptor or any protein, right. It changes the conformation of the protein, because protein has any specific conformation as it is. Then when the ligand or a small molecule they interact with any pocket, right, this will change the conformation, right, and this will also make to have the interaction between the protein as well as the ligand. This ligand can be a substrate, or this can be inhibitors or activators as well as neurotransmitters. Then how strong or what is the tendency of this specific ligand to interact with the protein, right, that tendency or the strength is called the affinity. So, we see affinity of a ligand. So, what is the meaning of the affinity of a ligand? The strength of the binding, right, how far this ligand can interact with the protein molecule, right, that will give you the affinity. So, if it is high affinity, what is the meaning of that?
Tightly bound.
It is tightly bound, right, like compared to low affinity. So, if you have several compounds then we can identify these different affinities of each compound, then we can find the specificity, right, among these different small molecules, which one has the best preference, right, to interact with a particular protein. So, if we see here ligand, generally it can be described with a Lipinski spread of 5, right. We can see if you see all the rules, and all the rules are the multiples of 5. So, there are 4 rules, all the multiples of 5. So, here this is Lipinski stated these rules for a drug, for a ligand to be kind of character of drug likeliness, right. Not necessarily all the drugs should follow this criteria, but most of the drugs, right, are a chemical compound which may act as a drug likeliness, they may have this type of conditions. The first one is the molecular weight. So, what is the molecular weight? You can see any compound, you can see the weight, right. The molecular weight should be less than 500 daltons. Then we have the hydrogen bond acceptor, the less than 10, and hydrogen bond donor less than 5. What difference between hydrogen bond acceptor and hydrogen bond donor? How to discriminate hydrogen bond acceptor and hydrogen bond donor?
So, hydrogen.
Yeah, hydrogen bond attached with the electronegativity atom, that is called hydrogen bond donor, right. Which one receives this one, that is called the acceptor. Then the hydrophobicity, that is mentioned as octanol water partition coefficient, right, that is not greater than 5. So, we have the hydrophobic, so it should not be not greater than 5. So, now if here this is the characteristic features of ligands. Now, when they try to interact, right, you can see a protein here. If we look at the protein, so you can see a pocket here, right. So, this is a pocket, this you can say a kind of cavity, this is probably a binding site. So, if the ligand tries to goes near to this protein, right, for example, here this is the ligand, here this make the complex. So, you have protein has a binding site, right, and the ligand can go to the binding site and they can interact and finally make the complex. Here you have to think about 2 different aspects. One is the pose, right. So, you have the cavity, right, acting as a binding site. What is the pose of this particular cavity? What is the conformation of that particular binding site? Depending on the conformation, then the ligand can have different efficiencies, right. If it is fit well, then it can tightly interact, right, in this case the affinity is very high, right, or if it does not fit well, then in this case the affinity is less. So, one is the pose, the conformation of that particular binding site, and the second one you can see the binding affinity, we can relate in terms of a score, which will give you the strength of the binding. So, 2 different aspects, one is the binding site or the pose, and the second thing is the scoring. So, what is the meaning of binding site or a pose, right? Here you can see this is the part of the protein where the ligand binds. In this figure you can see here this is the place where the ligand can probably bind, right. So, this we can say as the binding site, and generally this kind of a cavity on a protein surface, right, because this, the interior part is highly covered and they are mainly imparting the stability of the protein. In this case you can see generally type of cavity, right, on the protein surface. Then how to identify these active sites, right. So, generally if you look at the crystal structure of a protein, right, bind to this any node inhibitor, and if you take the inhibitor you can see there is a cavity, right, where the ligand bound with this protein. So, you can look at the structures, you can see this binding site, and also a lot of several computer programs available. In this case you can give the PDB structure, right, you can identify the binding site of any particular protein, right. The second thing is the binding mode, right. What is different binding mode? This will give you the scoring, right. So, we need the binding site, then the second one we need the binding mode or the pose, and based on that the ligand can bind and we can score whether the ligand can properly fit or not. So, what is the binding mode or the pose? This will give you the geometry of the ligand, right, in the binding site. So, geometry is terms as location, orientation as well as the conformation, right. You can see both in the case of the protein site as well as the ligand site. Ligand site also you can change the conformation and make various conformations, right. For example, if I see show this ligand, this one conformation. During these translation rotations we can change several conformation of this particular ligand, then you can see whether this ligand can fit with this binding site, right. Fine. So, now we have the protein we identified the binding site and we have the ligand we can also generate different conformations. Now, the next aspect is whether these two will interact or how far these pores of these different ligands they can fit with the binding site, right. For this we use the term docking, right. What is the meaning of generally docking? It is again the interaction between the two molecules, right, a dock between the two molecules, so for example the protein as well as the ligand. So, what the docking does? So, docking generally tries to find the best match. If you have a protein as well as a ligand, it will try to interact with the particular site with the best match. For example, you can see any protein complexes or the protein ligand complexes or the protein DNA complexes, right. It is a kind of lock and key mechanism. You can say shape complementarity, right. For example, if you have a binding site like this, and if here you can see the binding sites, right, then you can find the ligand which, right, you can see to accept, right, to interact with that particular place, like the shape of the shape complementarity, right. So, find the best match, right. So, then if you do like this, what will happen? The orientation changes which maximizes interaction between the protein and the ligand and minimizes the total energy of the complex. In this case, it gives a strength of binding, right. You can see various examples like protein nucleic acids, protein ligand, protein and so on. So, this binding is very important, and based on the strength of these interactions you can relate with the activity of the compounds, and this plays an important role in the structure based drug design to identify the lead compounds, right, to identify the inhibitors, right, for any specific targets, right, which are involved in these different pathways or in different diseases. Fine. So, how the term structure based drug design emerged, how they started to identify some compounds and how they related these small molecules or ligands, right, to interact with the proteins in the discovery of the structure based drug design. Earlier days they tried to check the drugs by trial and error methods, right. From the ancient days, if they are sick they take some leaves, right, and they eat some of the leaves. Even nowadays, right, so many medicinal compounds, right. So, mainly the compounds are important, but if you eat these leaves, right, and several other parts of the these plants, right, they are used for say potential drugs. So, they get the powders made from willow bark, for example, right, useful for the treating headaches, pains, fevers and so on in the earlier days. Then they try to get what are the active ingredients. If you eat some of the leaves, right, it can be medicinal, but what are the active compounds which one interact with the system, right. Then they found that 1980s they found that this is a close analog, right, then that became aspirin in 1899, right. So, they found that there is a chemical compound that is present, right, in these plants, right. This is close to the aspirin in 1989. Because of the birth of the structure biology, then when you get the protein structures, which structure first they determine the structure, which protein?
Insulin.
That is for the sequence. For the structure, that the hemoglobin, myoglobin, that they call the structures and got the Nobel Prize, right. So, when we got the structures, then we know the ligands which are working well as a drugs, then they use the structural biology, right, and then they see which ligands and how they fit with the structures. This gave this structure based drug design, right. So, we say given the 3D structure of any enzyme, right, you can use a structure based drug design, right, where the inhibitor can target, right, in a particular protein. For example, in this figure, okay, here if you see this is the protein and this is the ligand, right, how this ligand can fit with the protein, right, mainly the shape complementarity and kind of the lock and key mechanism, how they will interact, right, and see how the activity changes. Fine. So, I show an example. So, here left hand side like this is the database of small molecules, right. There are many databases available for small molecules. For example, a PubChem, you have zinc database, Chinese medicines, enamine database, right. There are many databases. Likewise, for different diseases, for example, chikungunya or dengue, right. Recently, this is going on, and many other viruses, right. So, there are several proteins which are involved, right, in these diseases, right. for example RNA dependent RNA polymerase, right, for the case of the dengue. So, in this case you can have the targets. So, now the question is, if you have the database of small molecules and we know the target, right, then how to identify the lead compounds which can fit with this target, and this could be useful as to design or get a drug molecule, right. So, we try to use different compounds, and in this case we use computer drug design. So, there are various aspects like docking, screening, QSAR, I will explain the details. So, using this information they will see some molecules, so this can be a drug, and other case this is not drug, right. Also the drug likeliness depends upon the molecular weight, heteromer donors, acceptors, as well as it should pass other trials, right, toxicity as well as the side effects, right, it should pass all the clinical trials. So, using bioinformatics, right, we can scan millions of compounds, quickly we can scan millions of compounds, and then we can provide the information that okay, these are the potential compounds, right, which could be used for the screening, right, and then from that screening we can identify the compounds, we can reduce enormously and see this can be used, right, for the further experiments, right, to see the activity as well as for the, other different animal studies. So, if you want to carry out computer aided drug design or structure based drug design, so what are the features, what are the input information we require, right. There are two different aspects. One, we need structure, one is a ligand. If the structure is known and the ligand is also known, then can we do this? We can do that, right. If you know this is here the known structure, right, and here the known ligand, right, then you can see these ligands can interact with this protein, right, you can see the docking and see whether this ligand will fit, right, with this particular protein, right, and this could be a probable lead compound or not. Now, the second question is, if we know the protein structure and we do not know the ligand, can we use structure based drug design? We can do, right, because we have a set of libraries for the ligands, protein structure is known. So, we can screen all these small molecules with respect to the protein, and then we can see okay, these are the probable compounds, right, which could be a lead compounds, right. It is a kind of the needle in a haystack to finding this one structure from a set of structures, right. We have pool of small molecules, and from that the set we have to identify the probable compounds. And the next option is, we know the ligand, but we do not know protein structure. In this case, can we use the computer drug design, right? If protein structure is not known, but ligands are known. In this case, if we unknown the protein structure, can we predict the protein structure? Yes, right. We discussed about various types of modeling. What are different types of methods to get the protein modeling? Homology modeling, ab initio, fold recognition, and the hybrid methods, right. So, if we do not know structure, we can get the structure, right, and the structures are known, then we can try to interact with the ligand and we can do that. But if structures are not known, ligands are not known, then can we do, right, but we can do CADD, but that is not accurate, but it will give mainly wrong results. In this case, CADD cannot be used, right, because we need at least some sort of experimental data, right. Without any experimental information, right, we can get some models, but that models are not reliable. In this case, we cannot trust this computer drug design to get the probable lead compounds. Fine. So, we look into the drug discovery. So, right, if you want to have a drug, normally how many years it will take to get a new drug? It is almost about 10 years or 10 to 15 years it will take, right, from the lead compound, and then do all these experiments, get the activities, then the animal studies, then you have to do with the several clinical trials, right, phase 1, phase 2, phase 3, then finally if you go with the several million compounds, you ended up 1 or 2 compounds, right, as a potential drug, but it involves lot of time, right, and also lot of efforts we need, and also involves lot of money, right, because it is very expensive. So, to do all these trials. So, if we start everything from this experiment, they do the wet lab experiment from the beginning, then it involves lot of time, right, it is a big process. So, to reduce the samples, to reduce the probable potential compounds, right, we can use the computer modeling, right. So, to see or screen the probable compounds which could be act as lead compounds. For example, we have the chemicals, say ligands with the biological system, right, it may interact with the different types of the proteins, right, and whether we give the desired response or not, right, it should work, right, but it should not give any side effects, right, and should not give any toxic, it should not be toxic. So, whether we can give, so we bioinformatics can do, right, to identify the initial set of compounds at least, right, we can identify these lead compounds. So, now we have the protein and we have the ligand, right. How to do the docking? Before doing dockings, we need to prepare the protein, also we need to prepare the ligand, right, because there will be a lot of issues in the case of proteins as well as the ligands, right. So, we have to work on that. So, let me explain what are the various options. So, how to get the PDB structures from X-ray crystallography? So, we will get the direct structure from X-ray crystallography? No, we do not get, right, we do not get the structures. What will we get? Map. We will get the electron density map. From the electron density map, we derive the structures. But how accurate the structures from the electron density map? If the map is not clear, then the resolution is very low. In this case, we are not sure about the atomic positions of this all the atoms, right. So, then second case, the water molecules, right, sometimes they put lot of water molecules, right, to crystallize and to solve the structures, right. So, in the case of the protein preparation, right, so if the water molecules are important, we keep the water molecule. If the water molecules are not important for the ligand to interact, then in this case we can remove water molecules. Second thing is, most of the PDB structures, if you see, the hydrogen atoms are missing, mainly in the case of X-ray structures. If you see the NMR structures, you can see the hydrogen atoms, but if you look into the X-ray structures, most of the structures the hydrogen atoms are missing. So, in this case, you have to add the hydrogen atoms, right, with all the missing atoms, because most of docking programs require the explicit hydrogens. Why do we need explicit hydrogen? At least for getting these hydrogen bonds are very important. So, in this case, we need this hydrogen to see explicitly where we can have these hydrogen bonds. So, we require the hydrogen atoms, we can add hydrogen bonds, but some many cases we can add, for example, the CH3 group. So, 2 or 3 hydrogens, we can see the positions, we can add easily. But if you say the case of the acidic and basic side chains, the question is whether this is charged or it is neutral. So, there is a confusion, this is ambiguity, so adding all these things. So, then the main aspect is, if you incorrectly assign the protonation states, then it will give the poor results. Well, the active sites, when the ligand interacts with the protein, so you have to assign the exactly protonate site. For example, if you take the glutamic acid or aspartic acid, either we can give COO minus or COH, but they are same or different? They are different, right. Here if you see the COOH, its OH is hydrogen bond donor, but this is not, right. So, we need to be careful to assign all these protonated states at the active site, right, because that is the place where the ligand can interact. Then if you take the histidine, right, it is a base, right, and it is a neutral form, right, it has either the base, right, so positive charge, or neutral form, it has 2 tautomers, right. In this case, we have to check which one is the preferred ones for the ligand to interact. So, what is the tautomers? These are isomers of organic compounds, right, they interconvert the chemical reaction, right, for example, from the keto to enol due to the migration of a hydrogen atom or proton, right, from the single bond and adjacent double bond. See here this H, this is CO, this is keto group. So, this protonations happen here, then finally this single bond is OH, this is called the keto enol tautomerism, right. So, what is the isomers? Right, they have the same chemical formula, but different chemical structures, right. So, now we need to consider mainly about these tautomers, specifically in the case of these histidines and the conversion of one chemical group to another chemical group. So, now the next aspect is, as I discussed earlier, the crystallography gives electron density, but not the structure, right. In poorly resolved crystal structures, it is very difficult. For example, see the asparagine or the glutamine. Here NH is down, but this O is up. It is also possible the other way around, right. If you change the conformation, then the ligand also the interaction will be different, right, because if the ligand we gave one pose, if the oxygen is this side it will have different interactions, if the other side will have different interactions, right. So, this orientation that is important. Then we have the we need to flip the amide or imidazole. So, mainly in the case of the histidines also we need to see where are these nitrogens. Then how to decide which one is the best, right. So, we can decide based on the structures with ligands. If you see the PDB, several structures you can give free structures, also many cases you have the structures with ligands. With the ligands you can look at the structures and see what are the probable orientations, probable conformation, and what are the probability of having these histidines, right, and which state. Then look into this information and then you can decide. For example, in your case, okay, this could be the most optimal one, fine. So, if you go to the protein side is fine, then go to the ligand side. Ligand side also we need to have a probable 3D structure, right, because in the docking mainly bond length, bond angle will not change much. So, they do not consider this, but torsion angles are important, right. So, torsion angles are important, that will give you the rotation around the different atoms. So, here also you have to see the protonation state and the tautomeric form, right. This will change or influence the hydrogen bonding ability, whether they have the strong bonds or the tight bonds or it is loose. So, here also we see the protonate state, right, either use a physiological pH and use single atomer or use all possible protonation states and then look at the lowest one, the highest score, then take that for the further processing. So, that you can do, right. So, now you have the protein, we analyze the protein and prepare the protein, then we have the ligand, ligand also we have chose the conformation ligand, right. Then what we can do, then we start docking.